Hello NEET aspirants, myself Madhu Yadav. I am a zoology lecturer at Sri Chaitanya Goshala, and today I am going to discuss with you the another part of the transport of gases. I have already discussed the transport of CO2 on this channel itself, so you can check it out if you have not watched that video. And after that, in continuation, today I am going to discuss transport of oxygen. After transport of CO2, I am going to discuss today transport of oxygen. So let me start without any delay the transport of oxygen. See, I have already discussed the transport of oxygen will occur in two forms. Transport of oxygen occurs in two forms via blood. Two forms. So one form will be the dissolved form, just like transport of carbon dioxide is also occurs in dissolved form. Transport of oxygen also occurs in the dissolved form. And this dissolved form, it travels via plasma, because plasma is the fluid part of blood. I have told you in the blood there are two things which are going to help us in the transport. One is plasma, another is RBC. One is the fluid part, that is plasma, and another is RBC. In the RBC, there is a hemoglobin which is going to play a great role in the transport of oxygen, right? So now, how much percent of oxygen will travel in the dissolved form? Only 3%. Only and only 3%. Whereas I have talked about CO2, 7% travels in the dissolved state in plasma. Why? Because CO2 solubility is more than that of oxygen, right? Now, in the second form, if we talk about the second form, that is oxyhemoglobin form. Oxyhemoglobin form. What is this oxyhemoglobin form? You can write it as HbO2, or you can also write it as one hemoglobin molecule binds with 4 oxygen. Then you can also write it as HbO8, right? But mostly people are writing like this. So it is simply oxyhemoglobin basically. In oxyhemoglobin form, it will travel via RBC because hemoglobin is present inside the RBC, right? Now, how much percent? So much of percent, 97%. 97% of the oxygen travels in this form. 97% of the oxygen travels in oxyhemoglobin form, right? I have already told you one hemoglobin molecule will take up 4 oxygen. To one hemoglobin, 4 oxygen can easily bind. And this binding of the oxygen, it is also very interesting. We will see how the first oxygen will bind and how then second, third and fourth will bind by the help of a curve. We will see that. But before that, let us start. Transport of oxygen will start from where? Transport of oxygen will start from the alveoli. As in the last session, we have seen the transport of carbon dioxide. It was started from the tissues. It ends at the alveoli. Here, transport of oxygen will start from the alveoli because here partial pressure of oxygen is high, partial pressure of carbon dioxide is low. Near to the alveoli, let us suppose this is the blood vessel. In the blood vessel, let us suppose this is the RBC. And here is the plasma, fluid part of blood. So, you can see plasma here and you can see RBC here. You can see RBC here. How much O2 will come into the dissolved form? 3% O2. This is the 3% O2. Rest of the 97% O2 will come. It will meet hemoglobin and it will form oxyhemoglobin. So here what is happening at the alveolar site? What is happening at the alveolar site? Association of oxygen with hemoglobin. There is association. It represents association of HbO2. Right. Now this blood will reach to the tissues, right? This is the same blood which I am making, and it will reach to the tissues because they require the oxygen, right? The blood has to give the oxygen to the tissues. So now let us draw here tissues. These are the tissues. In the tissues, partial pressure of oxygen is less because what is getting evolved? Carbon dioxide. So partial pressure of CO2 is more and partial pressure of O2 is less, right? Now, here in this blood, how much of oxygen is present in the dissolved form? 3% of oxygen is present in the dissolved form. Which blood is this? Oxygenated blood, because it has oxygen right now. So, this blood is coming near to the tissue. This is oxygenated blood. In the oxygenated blood, the partial pressure of oxygen is more, 95 mmHg. And in the tissues, the partial pressure of oxygen is less, that is 40 mm of Hg. Due to this partial pressure difference, here the hemoglobin will leave the oxygen. See, hemoglobin loves both the gases, oxygen as well as carbon dioxide. But wherever it will see the high partial pressure value of any gas, it will bind to that gas only. For example, wherever CO2 partial pressure is more, it will bind to CO2. And wherever oxygen partial pressure is more, it will bind to oxygen and it will dissociate from the CO2. That's what you have seen at the alveolar site when we have seen transport of carbon dioxide. When the transport of carbon dioxide will occur from blood to alveoli, in the alveoli, partial pressure of oxygen is more. So, HbCO2 dissociation and HbO2 formation. That's what? HbO2 formation. HbO2 formation. But at the tissues, when the CO2 partial pressure is more, hemoglobin is going to leave the oxygen and it is going to carry the carbon dioxide now. So, here what will occur? Dissociation of this HbO2 will occur and Hb will dissociate from the O2 and this O2 is going into the tissues. This 3% O2 is also going to the tissues directly, and this O2 after dissociating from the hemoglobin, it will go to the tissues and then tissues will be happy, right? They will be happy after getting the oxygen. So, it is very easy as compared to transport of carbon dioxide. I have already discussed in detail the transport of carbon dioxide with you, and now we are discussing the transport of oxygen. Very easy. Association of HbO2 at alveoli, dissociation of HbO2 at tissues. Dissociation of HbO2 at tissues. Now, both of these are represented by a curve that is known as oxygen dissociation curve. So, I will discuss with you the oxygen dissociation curve also in today's lecture. But before that, let me tell you how much oxygen is carried by how much ml of blood. See, there are some values like, do you know that in your 100 ml of blood, how much hemoglobin is present on an average? Let us take it, the value, let us take it 15 gram of hemoglobin is present. 15 gram hemoglobin is present in 100 ml of blood. And do you know that 1 gram hemoglobin can carry 1.34 ml of oxygen. 1.34 ml of oxygen. So, if somebody will ask you 100 ml of blood will carry how much of oxygen? If 100 ml of blood has 15 gram of hemoglobin and 1 gram of hemoglobin can carry 1.34 ml of oxygen, then 100 ml of blood can carry 15 into 1.34 ml of oxygen. That will be equal to nearly 20 ml. So, if somebody will ask you in 100 ml of oxygenated blood, how much oxygen is present, then the answer will be 20 ml. Now, the very important thing. The next important thing is to understand out of this 20 ml, out of this, under normal conditions, under normal physiological conditions, under normal physiological conditions, under normal physiological conditions, only 5 ml is delivered to tissues. Only 5 ml is delivered to tissues. So, rest of the 15 ml will be left in the blood, right? And that 15 ml is reserve. That 15 ml is reserve, right? So, whenever you are doing heavy exercise, you are running fastly and you are doing heavy exercise, so that is a strenuous condition. In that condition, your body requires more oxygen than the normal condition. So, here it will be different. Out of this, under strenuous conditions, how much ml of oxygen is supplied? 15 ml of oxygen is delivered to tissues. 15 ml of oxygen is delivered to tissues. That means 5 ml will be left in the venous blood. 5 ml will be left in the deoxygenated blood. After the supply of oxygen, the blood will become deoxygenated, right? If somebody will ask you, under normal condition, under normal physiological condition, how much ml of oxygen is left in the venous blood? Then 15 ml of oxygen left in venous blood, venous or deoxygenated blood, But if somebody will ask you, under strenuous conditions, how much ml of oxygen is left in the venous blood? Only 5 ml of oxygen is left. Only 5 ml of oxygen is left in deoxygenated blood. In how much ml of deoxygenated blood? In 100 ml. In 15 ml of oxygen left in 100 ml of deoxygenated blood. Here also 100 ml of deoxygenated blood. So total 100 ml of oxygenated blood has 20 ml of oxygen. 100 ml of oxygenated blood has 20 ml of oxygen. Why? Because 100 ml of blood has 15 gram hemoglobin and 1 gram hemoglobin can carry 1.34 ml of oxygen. So, if you will calculate for 100 ml for 15 gram, it will become 20 ml. This much oxygen is present in the oxygenated blood. But out of this, only 5 ml is delivered in the normal condition and 15 ml will be delivered in the strenuous condition, right? So, when 5 ml is delivered, the rest of the 15 ml will be called as reserve, right? That is why 20 ml of oxygen will be present in the 100 ml of oxygenated blood, right everyone? So, out of this, only 5 ml is delivered. This is written in NCERT and this is very important, and on the basis of this, questions can be asked. If in 100 ml, if it is telling you 100 ml is supplying 5 ml, then 200 ml will supply how much, or 1000 ml will supply how much? So, like that questions can be asked. So, this is important to remember. Now, I will discuss with you the very important oxygen dissociation curve, which will explain you the binding of oxygen with the hemoglobin, how it occurs. So, now the topic is oxygen dissociation curve. Oxygen dissociation curve. So, what the curve which you are seeing, this is the curve in the normal condition. This is the curve obtained in the normal condition, when in the body the conditions are normal. During normal condition, this is the normal curve, right? What is the shape of this curve? The shape of this curve is sigmoid. Why it is sigmoid? This is sigmoid due to cooperative binding. What is the meaning of this cooperative binding? See, what is the meaning of cooperation? Cooperation means help, right? when one oxygen is bound to one hemoglobin. See, in one hemoglobin, how many oxygen can bind? To one hemoglobin, 4 oxygen can bind. So, when to one hemoglobin, 1 oxygen will be bind, there will be 25% saturation. Because when 4 will be bound, there will be 100% saturation, right? So, there are 4 iron atoms, right? There are 4 units. There are 4 polypeptide chains in the hemoglobin structure, and inside each polypeptide chain, there will be a central metal ion, that is iron. So, hemoglobin is an iron containing pigment. To that, the oxygen will bind, right? So, it will make oxyhemoglobin. So, when one oxygen is bound, then it will call the others also that only 3 seats are left, you have to come fast, right? So, the another oxygen will come. Then the 2 which are bound, they are calling the third one. They are helping the binding of the third one. And when 3 are bound, all the 3 are helping the binding of the fourth one. That is the cooperative binding, and that is why we get this curve as a sigmoid curve, right? We get this curve as a sigmoid curve. In this sigmoid curve, you can also say S-shaped curve. You can see in the starting, the saturation is taking time, and then it is occurring fast. And here you can see the 100% saturation. This is the 100% saturation of hemoglobin with oxygen, and this is the partial pressure at which 100% saturation has obtained. So now if you will see, this is the 50% saturation. This is the 50% saturation. 50% saturation is achieved somewhere here, right? 50% saturation is achieved somewhere between 25 to 30. Somewhere between 25 to 30 mmHg partial pressure, 50% saturation of hemoglobin is achieved under normal conditions. But we will see the curve will be shifting to the either directions, to the left direction or right direction, right? Towards the right direction or towards the left direction. So now under which conditions we will see the shifting in the curve? Let us see that. Let us suppose this is the right shift. Yeah, this is left. This is left shift. The white one I am making is the left shift, and the blue and the yellow one which I have drawn is the right shift. So you can see this value. See, when the curve is normal, I have told you this value between 25 to 30 mmHg partial pressure, na? We will get the 50% saturation of hemoglobin, right? This value is known as P50 value. P50 value. What is P50? P50 is the partial pressure at which, it is the partial pressure of oxygen at which hemoglobin is 50% saturated. You can see that it is going to change when the curve will shift to the left or when the curve will shift to the right. Yes or no? When the curve will shift to the right, P50 is going to increase, and when the curve is going to shift into the left, then the P50 is going to decrease. What it is representing? It is representing that, what it is representing? It is representing that here the affinity of the hemoglobin towards the oxygen is more. That is why 50% saturation is occurring quickly, right? At less P50 only. But here the affinity is less. That is why saturation is occurring is achieved at a high partial pressure. That means it is not achieved actually. This is not the oxyhemoglobin formation. It is actually the oxyhemoglobin dissociation. So let me tell you what it is representing. This is the left shift. This is the left shift, and the yellow one is the right shift, right? It is the right shift. So right shift, in the right shift you have seen increase in P50. I have talked about increase in P50. It is also known as Bohr's effect, and I have talked about Bohr's effect in the previous lecture of transport of gases. I have talked about two effects, Haldane's effect and Bohr's effect, in the previous lecture of transport of gases, right? So basically, in the Bohr's effect, I have told you there will be dissociation of oxygen from hemoglobin. And where the oxygen is dissociated from hemoglobin? At the tissue site, because tissues only they require the oxygen, right? So that means we are going to see the shift in right at which level? The shift in right is seen at the tissue site. At the tissue site, it will take place at the tissue site, whereas left shift will take place at alveolar site. So now let us see the factors which are responsible for these shifts, right? The important factors which are responsible for these shifts. And this left shift is, now you can write that it represents what? It represents association of HbO2. Association will occur when partial pressure of oxygen will be high. If partial pressure of oxygen is high, then partial pressure of carbon dioxide is less. If CO2 is less, then hydrogen ion concentration will also be less. You have seen in the transport of carbon dioxide that when CO2 is coming into the blood, there is release of H plus in the blood, right? So CO2 is less, H plus is also less. If H plus is less, then pH will be more. If pH is more, that means it is not acidic. Acidity is also less, right? These are the factors, and one more factor is there, that is known as bisphosphoglycerate, that is also less at the alveolar site. It is more at the tissue site because it is an intermediate of glycolysis. Glycolysis is a part of cellular respiration. In this glycolysis, the glycolysis is occurring, so many substances are getting formed, right? And then this compound is getting formed, that is bisphosphoglycerate or diphosphoglycerate, BPG, or it can be called as DPG. So obviously, when cellular respiration is occurring at the tissue site, BPG or DPG will be more at the tissue site. How it will favor the dissociation of HbO2 and how it will favor the association of HbCO2, that I will explain you. So these are the factors which are responsible for the left shift. High PO2, less PCO2, less H plus ion concentration, high pH and less acidity. Obviously, when H plus will be low, then obviously pH is going to be high and acidity is going to be low, right? It is dependent upon the hydrogen ion concentration. Now let us talk about the right shift. At the right shift, at the tissue site, you know that what will happen? Dissociation will happen. So it represents dissociation of HbO2, dissociation of oxyhemoglobin, formation of HbCO2, that will occur when partial pressure of oxygen will be less. Exactly opposite. Partial pressure of carbon dioxide will be more. If CO2 is more, then H plus ion concentration is also more. When H plus is more, that means pH is less. When pH is less, that means acidity is more. Yes or no? The value of pH, when it will be very less, pH value 2, 3, it is a strong acid, right? H plus ions are very high. And the BPG or DPG is also high. This favors, you can write here, it favors carb-amino-hemoglobin formation. That is why it is responsible for dissociation of HbO2. Just like hemoglobin has site for oxygen, has site for CO2, Similarly, hemoglobin has also the site for BPG, bisphosphoglycerate. And if this is bound to the hemoglobin, then it is a friend of CO2. It will tell the CO2 that you come with me. है ना? So it will favor the binding of CO2 with hemoglobin. That means it will dissociate HbO2, hemoglobin and oxygen dissociation. That's what is occurring at the tissue site. So if somebody will ask you, decrease in P50, left shift, increase in P50, right shift, Bohr's effect is seen at right shift only. Increase in P50 is only known as Bohr's effect. Due to the increased carbon dioxide partial pressure, there will be increase in the P50 value and it is known as Bohr's effect, right? Right shift will be seen at the tissue site and the factors responsible are these, whereas left shift will be seen at the alveolar site and these will be the factors. I hope you have understood oxygen dissociation curve. First association will occur and at the end when dissociation will occur, then only our tissues will get the oxygen. So oxyhemoglobin is not a stable compound. It will form and it will break. If it will not break, then how oxygen is going to be taken by the cells, right? So this is all about transport of oxygen. Now on the basis of transport of oxygen, let us discuss some questions. So you have to identify the wrong statement. What is the wrong statement with reference to transport of oxygen? Just now we have seen the transport of oxygen. Partial pressure of CO2 can interfere with oxygen binding with hemoglobin. Absolutely correct. Yes, it can interfere. Higher H plus ion concentration in alveoli. So you have seen in alveoli, H plus concentration is not high. In the alveoli, what is happening into the alveoli? In the alveoli, association of HbO2. In the alveoli, PO2 is more. In the alveoli, PCO2 is less. If PCO2 is less, then H plus ion concentration is also less. Right? So it is a wrong statement. Let us see the other statements. It is a wrong statement, beta. Let us see the other statements. Low, it is PCO2. Low PCO2 in alveoli. Yes, it is correct. Low PCO2 in alveoli favors the formation of oxyhemoglobin. Right? Binding of oxygen with hemoglobin is mainly related to the partial pressure of oxygen. Yes, absolutely. When partial pressure of oxygen will be high, it will be binding with the hemoglobin. So, other statements are correct. Only the B statement is incorrect. You can see here, you can verify here. Higher H plus concentration is not there in the alveoli. I have shown you in the alveoli. At the alveolar site, there is low H plus concentration, beta. If there is low H plus concentration, it is the wrong statement and this will be the answer. Going to the next question. Select the favorable conditions required for the formation of oxyhemoglobin. For the formation of oxyhemoglobin at the alveoli, PO2 should be high. It is correct. PO2 should be high. In the low PO2, oxyhemoglobin will not be formed. When high PO2, then PCO2 should be low. Yes, correct. But here, PCO2 should not be high. So, it is incorrect. So, if it is correct, then this is going to be your answer. When there is low PCO2, there is low H plus. When there is low PCO2, there is low temperature. Yes, absolutely. CO2 is related with the temperature. When at your tissues, CO2 is evolving more, the temperature will be more. So, CO2, wherever it is more, temperature will be more. So, you can add this. You can add this here. Temperature. You have already written PCO2 is less, then temperature will also be less here. Whereas in the right shift, the temperature will be more. The temperature will be more because here CO2 is more. It is occurring at tissue site. Right, everyone? So now you understood the last question. This question you understood? Favorable condition. So like that, they can ask you the factors upon which the right shift and left shift. You have seen the factors which are linked with the right and left shift. I hope all this is clear to you. I hope you have understood the transport of oxygen very well and the oxygen dissociation curve. Right? It is very important, and please read NCERT. After watching this lecture, go through the NCERT and try to understand everything and solve the relevant questions. Thank you, one and all.